Set, girl.
So, what is artificial blood? Artificial blood is an innovative concept of transfusion medicine that is specifically designed to perform the task of transport and delivery of oxygen in the body, and also to replace the function of human blood transfusion. Several molecules of blood have been developed progressively to achieve the objective and continuously made to get the ideal blood substitute. This artificial blood does not require compatibility testing, free from blood infection, has prolonged life, and does not require refrigeration.
There are two types of artificial blood. The first one is perfluorocarbons, PFCs. It is an entirely synthetic inert molecules containing fluorine and carbon atoms. It is capable of dissolving many gases including oxygen. That's why it can carry more oxygen than red blood cells do. The second one is hemoglobin-based oxygen carriers, HbO6. It is found in the blood as the natural oxygen carrier and may be extracted from several sources such as human red blood cells from outdated blood and red blood cells from cow's blood.
Next, we'll move on how artificial blood is made. Artificial blood can be made through many ways such as synthetic production, chemical isolation, or recombinant biochemical technology depending on the type of artificial blood. For the first type, which is hemoglobin-based oxygen carriers, hemoglobins are synthesized and isolated in the lab. A strain of E. coli bacteria that has the ability to produce human hemoglobin is used to help synthesize hemoglobin. The protein is harvested and then fermented until the population is great enough. After that, it is transferred into a seed tank, which is a large stainless steel kettle that provides an ideal environment for growing bacteria until enough time has passed. Next, the product is transferred into a fermentation tank where it is filled with a growth media needed for the bacteria to grow and produce hemoglobin. After there is enough hemoglobin, the tank is emptied to start the isolation process. After the isolation process is completed, the synthesized hemoglobin are then modified at a molecular level. They are modified by its structure or chemical composition. This is to enhance stability, function, or other characteristics. Lastly, they are reconstituted back into an artificial blood formula, combining the modified hemoglobin with other components to ensure its function as a blood substitute. This includes combining them with carriers, stabilizers, and other elements. In order to ensure stability, oxygen carrying capability, and compatibility with circulatory system. Various physical and chemical properties of the finished product are checked, such as pH, melting point, and moisture content. Next, we'll move on to the second type, which is perfluorocarbons, or known as PFCs. The PFCs are emulsified in water using an emulsifier. Emulsification is the process of mixing two or more immiscible liquids through building microbubbles. It generates thermodynamically metastable but kinetically stable mixtures. As PFCs are extremely hydrophobic, emulsifiers such as lipids, fluorinated compounds, tensides, or proteins are needed to bypass the immiscibility with aqueous media such as blood. Emulsifying agents help disperse PFC droplets in liquid. Then stabilizers are added to avoid the fusion of the droplets and maintain stability. Next, the properties of PFC emulsion are analyzed through its droplet size, stability, and oxygen carrying capacity. Other than that, additional components such as electrolytes and buffers are added to stimulate the conditions of natural blood. Finally, the artificial blood will be evaluated based on its capability of carrying oxygen and performance under various conditions.
Effectiveness and challenges. The effectiveness of artificial blood are it can deliver oxygen faster, minimize injury to the tissue, and universally match to all blood types. The challenges are it's not a true blood substitute, it can affect thin blood flow, and may cause hemosiderosis and chronic
The differences between real blood and artificial blood. For the first aspect is composition. For the real blood, it is composed of various elements including red blood cell, white blood cell, platelets, and plasma. While for artificial blood, it is typically contains synthetic molecules that can carry oxygen similar to hemoglobin in red blood cell. For the second aspect is the blood type. For the real blood, there are four types of blood, which is A, B, AB, O, and Rh factor positive or negative, while for the artificial blood is designed to be universally compatible. Lastly is the oxygen transport. For the real blood cells in real cell blood contain hemoglobin, a protein that binds and carries oxygen to tissue and organs, while artificial blood is hemoglobin also carries oxygen, but the structure and properties of the artificial hemoglobin may differ from natural hemoglobin.
In conclusion, artificial blood or fake blood has many advantages, but it still lags behind in real blood replication in terms of its properties and structures. Thus, more research is needed in developments of artificial blood to ensure potential benefits for future generations.